Our main base of operations is in the Florida Keys. We've been working at this site for about 10 years, and students go down with me at least once a summer probably to do some field research and do some field-based work. And they do experiments out in the flats or out on the reef, and then we bring samples back, we bring tissue back, we bring all kinds of stuff back with us, and then that's what they're working on right now.
A lot of work, because I'm going to the Florida Keys flats every day, collecting like different sponges and like algae to analyze. So it was a lot of work, hot, tiring, but sometimes fun because I saw things that a lot of people don't get to see. I got to like snorkel in like a coral reef, and not some people get to see that. I saw sharks, swim with sharks. It was just like a once in a lifetime opportunity, and it's just really humbling experience to see how beautiful the world actually is.
This is my third summer doing research at Richmond, and my second summer with Malcolm. We spent the first 2 weeks in Florida, and we collected samples. I did an experiment where I infected sponges with these chemicals that inhibit photosynthesis, and that's important because the algae in the sponges need photosynthesis to survive, and we think that they might be giving some of that energy from photosynthesis to the sponge. So when we affect their ability to make this energy, we want to see what happens to the algae in the sponge. Does it get eaten? Does it die? So that's kind of what my experiment's looking at right now, what I was doing in the lab. So after we did this experiment, we take the sponge samples and then we look at how many algae cells are in them. And so that's what I was doing on the microscope. You do Zoe's counts and count the number of cells, and then we're also looking at, under the microscope, what the algae cells look like now after they weren't able to do photosynthesis.
I'm working on like a hypothesis where it talks about how Symbidium, which lives in like sponges, have like a relationship with the Symbidium. Symbidium is algae, and they live within sponges, and they rely on sponges to provide for them the energy and protection from like things that eat them outside of the sponge. So it's kind of like seeing how those two things relate and how climate change impacts Symbidium, and like how sponges need Symbidium to survive. Without sponges, the whole ecosystem collapses. It's because they're keystone species. So just answer questions to see how they relate to each other.
One of the things that we're working on is a symbiosis that occurs between animal hosts and an algal symbiont. And the algae are actually important for coral reef development. In fact, all the coral reefs around the world rely on this symbiosis, and so we study it from a slightly different perspective. We use sponges as a model organism to look at in terms of host-symbiont interaction, and so everybody in the lab is working on different aspects of that research, including freshwater symbioses as well. So folks are looking at using molecular approaches and microscopic approaches, and we do some straight-up ecology, evolution, but yeah, everybody in the lab is working on different aspects of this symbiosis. The work they do here is different than the work they would do in a classroom. They get to apply the knowledge that they've been learning in a theoretical context in the class, but then they get to apply it to real-world problems, and they contribute to some really important research.
Not many people have this opportunity to do what I do, and it's kind of awesome about being at Richmond. They provide opportunity for undergrads to do a lot of things. It's like the best of both worlds. Like you said, it's just, you have great Division I sports, at the same time you have a great a career experience and opportunities. So that's why, that's one thing I love about University of Richmond. It's one reason why I came here. Is just like I can balance the two. I mean, it's tough doing both, but it's something I think a lot of undergrads miss out on.
I have the resources to really push myself harder than I could at home, and at the same time I'm getting research experience. I guess the worst part of my experience at Richmond is I have too many opportunities, so I'm trying to juggle soccer and research and all these things, and everything you can do at Richmond, like, your opportunities are limitless. Like, if you want to do research on diseases, you talk with a professor, they'll make sure you can do it. If I'm interested in Malcolm's lab, he'll make sure I can do it. But then the coaches are supporting me in this and the sport, and then I can do the sport I love and also get this real-world experience. I think that's the reason I came to Richmond, is because you can do all of this at the same time, and I was really dead set on a really good school academically so I could push myself and get this experience, but also be able to play the sport I love, and that's what I got at Richmond.